Now, typically when one thinks of in vivo optical imaging being applied to preclinical research, one envisions an image such as this, where you have optical data being superimposed onto the white light image of a mouse or rat, or onto the X-ray of a mouse or rat, for the purposes of orientating the location of that optical data. It should be emphasized, however, that there are many other animal model systems and constructs by which in vivo optical imaging can be applied to great use. My general rule of thought is that if you have a point source of light that emits from a centimeter or less from the surface of tissue, and you can get the object into the imager, well, then you have a model. And so here's a sampling of some of the many non-mouse model systems in which optical imaging can be applied. We're looking at transduced zebrafish here, as well as caterpillars that are transduced to express Rhinelin luciferase. You can have any number of bacterial cell lines that have been transfected to express a fluorophore. We also have many investigators that, before going in vivo, will evaluate their cell line or their nanoparticle construct that expresses bioluminescence or fluorescence light in an in vitro context to make sure that the amount of light being generated is going to be adequate for their purposes when going in vivo. It also provides them the opportunity to evaluate dose levels before going into an in vivo context. Now, there are investigators that are focusing on agrarian questions that will go ahead and make use of in vivo optical imaging as well. And what we're looking at here is the use of optical imaging to evaluate pathogen burden on produce going to market, literally, and the appropriateness of various different techniques to reduce this bacterial load. And be it cantaloupe or chicken wing, the results of this kind of work can be critical in setting up the appropriate treatment of produce in the process of harvesting and handling before it goes to market. Now, in the top right-hand corner, left-hand corner, what we're looking at here is an example of Cherenkov imaging. And first, a bit of science. So Cherenkov imaging basically deals with the use of high energy positrons that are being emitted by radionuclide. So think of PET imaging. And so if you have a PET radionuclide being used in your mouse model system, you can monitor and detect its biodistribution by using optical as a preliminary evaluation of your PET model. So how does it work? Basically, you have the positrons that are emitted by the radionuclide interacting with tissue electrons. And these electrons and positrons come together in an annihilation event. And from that are high energy photons that are generated. These high energy photons are traveling at speeds greater than visible light through the tissue. And as such, they leave a wave of blue light. Now, this is a surface biased application, as we know, because blue light does not travel far through tissue. That being said, there are a variety of applications for which Cherenkov imaging has its purpose. Now, finally, what we're looking at down here is simply the use of our sister imaging modality, the X-ray, for the purposes of evaluating the details of the structure of certain materials of interest to investigators. So this is an application that some of our investigators use when they make use of our imaging systems. So from amongst all of the many applications that have been testimony to in literature surveys, we're going to go ahead and just present some of the more common and highly valued applications of in vivo optical imaging when it comes to small animal preclinical research. And it should be mentioned that all of the papers that are presented here are peer-reviewed publications, and all the investigators are folks that are making use of SI imaging devices, either an AMI-HT or a LIGO imaging system. Now, in this initial sample of a publication image, we're looking at the central, most commonly used aspect of in vivo optical imaging when it comes to preclinical imaging research, and that is the early onset detection through high sensitivity monitoring of the cells of interest. In this particular case, we have a Wistar rat that has been intracranially injected with glioblastoma cells to be studied for their sensitivity to therapy. And they've already been transduced to express luciferase. And you can see from as early as day 8 onward, the cells are detectable. And as the cells grow, the image here would suggest that the cells are growing out to day 20 as the signal increases in intensity. Now, in this second study, which is looking at the ability to regulate the occurrence of metastatic lesions from a primary tumor, the investigators essentially wanted to be able to monitor with their optical in vivo imaging the occurrence and growth of the primary tumor site, followed by any incident metastatic lesion. And that was achieved here. What you can see at week 1 through week 4 in this particular mouse is the growth of the primary orthotopic breast tumor mass, as indicated by increase in signal intensity. And then on week 4.5, there was the occurrence of a metastatic lesion in the pulmonary space of this mouse. I've gone ahead and included some of the efficacy data from this study. They used a range of T-cell therapies for this control of the breast tumor mass, along with doxorubicin. The essential point that I'd like to make here is that in monitoring efficacy, they used mean tumor volume, which is fine and traditional, but they very easily could have gone ahead and start evaluating the overall efficacy of the different treatment paradigms by looking at overall whole mouse signal intensity. Now, the interesting aspect of this next study is that the investigators structured their experiment to assess the possibility of conducting the following clinical protocol. Would it be possible to label a glioblastoma tumor mass with a fluorophore reporter such that at the time of surgery, at the surgical site, the surgeon could use an excitation light to light up the glioblastoma tumor mass, courtesy of the fluorophore reporter that was directly bound to it? Now, as a preliminary criterion, the investigators realized that they would have to be able to successfully deliver this fluorophore to the brain space across the blood-brain barrier. So they went ahead and developed a particular micelle nanoparticle construct that was designed to do just this. And by way of a preliminary setup, they went ahead and challenged the test mice with a human glioblastoma cell line that had already been transduced with luciferase, so it would give a bioluminescent signal. And using the bioluminescent signal, they monitored the intensity of that to the point where they believed that there was an adequate glioblastoma cell mass. And you can see that point in time right here. The investigators then went ahead and used a 4 nanomole quantity of the fluorophore-loaded micelle structure, illustrated here, and introduced it into the mice via tail vein injection. Now, 24 hours post-challenge with the loaded micelle nanoparticle construct, the investigators went ahead and imaged both the control mouse, which had no glioblastoma, and the 4 test mice that did. And sure enough, what you can see here is that there is above background signal of the near-infrared fluorophore in the brain space of the 4 test mice, certainly of the 3 middle ones. And what the investigators did is they followed this over a 96-hour time course and saw a persistence of this elevated signal in the brain space area of all 4 of the test mice at the 96-hour time point. What I really like about this study, however, is that they went ahead and did one additional thing, which was to go ahead and evaluate not only the in vivo biodistribution as indicated by fluorophore signal, but to do a end of study necropsy and look at the biodistribution across all the major organs of interest. And sure enough, here, with the absence of tissue attenuation, the ex vivo evaluation, they saw again that there was elevated signal in the brain of the test mouse, demonstrated here. And by comparison, the brains of the 4 test mice versus the control mouse were notably brighter in fluorescent intensity, again indicating that not only did the nanoparticle direct delivery to the brain space, but because of the glioblastoma presence in the brain, that persistence of the near-infrared fluorophore signal in the brain was due to the glioblastoma. Now, in this next study, the investigators really want to accomplish 2 things. They wanted to be able to demonstrate that they could effectively detect the bone metastatic lesions of a breast tumor cell line. And secondly, they wanted to be able to evaluate whether or not their new novel polyclonal antibody against the CSF1 cytokine CSF1 receptor complex, which is known to promote unregulated cell growth within the breast tumor cell line, if that antibody applied to the mice in vivo could effectively suppress the development of bone metastatic lesions of this breast tumor cell line. So they evaluated 3 different treatment paradigms to determine whether or not, 1, they could see the breast tumors in their metastatic foci in the bone, and 2, to see if there was any specific reduction courtesy of the polyclonal antibody against the CSF1 cytokine receptor complex. And what you can see here is in the no treatment group out to day 31, there's a very nice progression of the formation of bone metastatic lesions after intracardiac challenge of the breast tumor cell line to the tune of 100,000 cells. And this is a standard way of initiating the metastatic lesion phenomena of primary breast tumor cells. And in the control antibody setup, you can see that there's a similar progression of bone metastatic lesion formation. It starts a bit later, but it's definitely there by day 31. And in the test mice, which were given the specific polyclonal against the CSF1 cytokine and receptor complex, you can see a variable outcome where the bone mets are formed in one mouse out at day 28 and day 30, but are really not seen at all in some of the mice, and one presented here being mouse number 2, where there was no metastatic lesion formation out to day 31. So in summary, this is a success in 2 ways. One, in that you see the ability to detect metastatic lesions in the bone microenvironment quite nicely. And secondarily, you see the moderate effect of the novel therapeutic in the ability to reduce the instance of metastatic lesion formation. Now, to further evaluate the precise location and extent of bone loss in the various different test group of mice, the investigators went ahead and made use of micro-CT. And what we can see here in one of the control mice, at the point where optical data indicated a metastatic lesion, there is progressive bone loss over the time course of the study in the head region of the tibia, close to the knee joint region. And in a more comprehensive evaluation of bone loss in the various mouse treatment groups, what the investigators observed was that for those mice that were given either no treatment or simply control antibody, there was indeed progressive bone loss in the femur or tibia bones close to the knee joint region. And by contrast, those mice that had been given the test antibody against the CSF1 cytokine receptor complex experienced no observable bone loss by micro-CT. So the general imaging application takeaway message here is that optical and micro-CT can function as complementary imaging modalities. where optical functions as a high sensitivity, rapid screening imaging modality for the purposes of detecting the cells of interest, in this case, the metastatic breast cells in the bone space of mice. Micro-CT can be used as a follow-up high-resolution imaging modality for the purposes of the exact characterization of the bone loss associated with these metastatic lesions. Now, finally, I should mention that SI imaging facilitates this complementary use of optical and micro-CT imaging through a commercial collaboration that we have with a company called Molecubes Inc., which makes a benchtop micro-CT unit. They make SPEC and PET as well. And we provide a universal animal bed that allows our LOGO imaging system users to do optical imaging in the LOGO and then transfer the animals to the Molecubes CT unit for further CT imaging. So in this next study, the investigators successfully used non-invasive in vivo optical imaging to demonstrate the relative efficacies of different drug treatment paradigms against their disease model of interest. Now, what were they studying? They were looking into the treatment of a particularly aggressive form of T-cell acute lymphoblastic leukemia that had been observed to express elevated quantities of PIM protein kinases. So they developed a PAM-PIM protein kinase inhibitor in the form of AZD. And they went ahead and evaluated here its in vivo efficacy. So briefly, the treatment was to take 200,000 cells of this particular strain of T cells known to express elevated PIM protein kinases that had already been previously transduced with luciferase, inject them via tail vein, wait for 3 days, and then proceed to treat them. The 20 mice that were so challenged were randomly grouped into 4 different treatment groups and were either given simply vehicle on day 3, AZD at 30 mg per kg, pontinib. Pontinib is another known multiple protein kinase inhibitor that they thought that they would evaluate in relative efficacy terms to their new test therapeutic, AZD. And they did combinatorial therapy as well. So in evaluating the relative intensities on a whole mouse basis of these mice in the different treatment groups, it became quite evident that there was some marginal reduction by AZD, again, some additional further reduction by pontinib, but by far the greatest reduction by the combinatorial therapy of both AZD and pontinib given together. And this is demonstrated graphically here. So one of the nice aspects about this paper was that the investigators didn't stop with the initial efficacy results. They went one step further. What they did is evaluated the efficacy of the same drug treatment paradigms against an engraftment model in which drug therapy didn't start until 14 days after challenge instead of the initial 3 days. So what did they see? Well, after 21 days of therapy, on day 35 of the experiment, they saw analogous results where the vehicle mice were heavily burdened with leukemia. The AZD treated mice, marginally less so, and the pontinib mice even more so reduced. But again, it was the combinatorial therapy of the AZD and the pontinib together where they saw truly a significant reduction in the overall leukemia burden in the mice. The unfortunate news here is that none of the mice survived. There was an extended lifespan for the AZD pontinib treated mice, but they too had premature death. The takeaway here from this study is that through the use of non-invasive in vivo optical imaging, the investigators were able to clearly discern differences in levels of efficacy by the different drug treatment paradigms tested. In the final application study to be presented here today is an example of virally mediated oncogenesis, where the investigators use a luciferase optical reporter to monitor the extent of the tumor progression in their mouse model system. So what exactly did the investigators do here? Well, they wanted to induce the formation of a soft tissue sarcoma, and they used a lysogenic viral particle construct to do this. Specifically, they used adenoviral Cre recombinase that had been modified to express interrupter sequences, if you will, that would prevent the effective transcription of two important growth regulatory proteins, TERP53 and P10. This adenoviral particle also expressed the luciferase gene, so that any kind of successful transduction of the muscle cells by the viral particle would be monitored through the light reporter function of luciferase. A secondary aspect of this study was to determine the best route of challenge to successfully generate the desired sarcoma. And what the investigators found out by testing either a subcutaneous or intramuscular challenge is that both routes were equally effective in generating sarcoma, as observed by the reporter function of luciferase at week 17. So the essential takeaway from this model is that it is readily possible to go ahead and induce cancers of interest in mouse model systems using virally mediated oncogenesis techniques. And furthermore, that this process can be monitored through the use of luciferase as an optical reporter.